Thank you, Tom. Thank you, colleagues and friends, ladies and gentlemen, and also those on the online to join this workshop. This is going to be an interactive workshop. In the first hour, we're going to give you the introduction, as well as we will update you about our progress and also share some of the preliminary results. And then in the second part, after the tea break, then we're going to have an open discussion about the next phase of this GEM UN Decade Program. And also we will also touch on what kind of solution we can offer to reduce pollution in the world in the third part of the discussion.
Wait. You can try now.
Oh, okay. Working now. Okay. So I want to highlight this. This program was inspired by the Global Monitoring of Pharmaceuticals project led by Professor Alison Boswell and his postdoc John Wilkinson. Back in 2018, he has this idea that we should use the same method to monitor pollutions in rivers around the world, because different laboratories use different methods, and also they will select different target compounds. It ends up the result cannot be compared. It's not comparable. So it is important everyone use the same method. So he came up with a simple idea. Why not he developed sampling kits and standard method? Everyone follow, and then they just need to return the sample to his lab in York University to do the analysis. And all the samples will go through the same process, making the result comparable with this fairness. So in this project, he involved 127 scientists, including myself, covering over 100 countries, and then covering 1,000 sampling sites in 258 rivers. And the results really interesting. As you see, over 1,000 sampling locations, almost all the sites except 2, all with medicines. That means the pollution of these pharmaceuticals is really widespread in the whole world, okay? Except 2. One is Iceland, and then the other one is in South America, in those rural areas. And the second phase, he found that this project find most polluted rivers actually in South America, Africa, Asia, those 3 regions, okay? And then the top 10, including Bolivia, Pakistan, Tunisia, Ethiopia, India, Congo, Kenya, Albania, Israel, and Costa Rica. So those are the top 10 in terms of the total concentration of the pharmaceuticals. What we find, it has a huge implication for pollution management. First, we find, as you can see in this diagram, the total pharmaceutical concentration, and this is the national income per capita, okay? As you can see, most polluted is in the second group, lower to middle income country. There are over 230 countries there. As you can see, their concentration is much higher. What does it mean? They have money to buy medicines, but there are no regulation. There are no green infrastructure to treat the waste and wastewater water properly. For example, what we find in Africa, many countries in Africa, they will make an open dumping site for waste, sometimes open burning of waste, and also they will make a pond, a big pond like swimming pool. They dump all the wastewater there, okay? But you can imagine, after heavy raining, all these pollutants just spill over into the river, okay? This is not very ideal situation. And if we want to clean up the pollutants for the whole world, we should target to this group of countries first. Do you agree? Right? If we can reduce like the other country, then we already remove a great deal of pollutants. Second things I want to highlight is that we also find the pollution level go hand in hand with this, the life expectancy. If people have a longer life expectancy, they tend to use more medicines because when we get old, we will have more problem in our health, then we have to take more medicines. That's common sense. Then the second one we find, it also go opposite to the poverty, okay? So for poor country, the poor they are, they have more pollution. It is because they cannot afford to build the green infrastructure to treat the waste and wastewater. That is a very obvious reason. And one thing very interesting we found is that the type of pollutants also different between poor country and the developed country. For example, in poor country, we found mainly painkiller and also antibiotics. But in a more wealthy country, we found very interesting. They have more antidepressant, means people not very happy. And also they have more diabetic bus in the river, as others, okay? And those are very interesting observations. One thing I didn't put down, you know, which country drink more tea and coffee based on the caffeine in the river? You can guess which country drink most tea and coffees. Based on our results, the answer is India. The second one is China. So those are interesting results we can find from the data. But most important highlight is that 36% of all these 1,000 sampling sites with medicines exceeding the safety limit. What does it mean? I give you an example. The blue bar means the river with exceedance for these chemicals. The blue one is for antibiotics. So in Asia, among the 13 antibiotics we measure, 8 of them exceeding the safety limits that can trigger antibiotic resistance in bacteria. That will eventually translate into superbug that will kill us. Okay? So the situation is not good, especially Asia, Africa, and also South America. You can see many blue bar. Okay? So because of this very important study, then we put all 127 all together to publish this work. Then after 1 year, we received more than 600 citations. Then we won the best paper award from PNAS. Then the citations still keep going out. And we think this is the best model. We can review the pollution situation around the world. Then I use this platform to apply for the UN Decade program in the first one back in 2020. Then we were luckily selected as one of the first phase UN Decade project. Then we spent almost 18 months to develop the methods and also prepare the sampling kits. Then we launched the program last summer, July. And I think how many of you are our members in this program? Already sending us the sample from Thailand, from Malaysia, and also Malaysia as well. So thank you for your support. We need more people to join us. So after today, please just register as our partner. We will do the same. We'll publish together with 100 people, okay? In this program, our founding member, including Adam Stoposo, Brian Brooks from US, Martin Duprin from Australia, cover different continents, and also Professor Wang Jueying from China National Marine Environment Monitoring Center, and also one representative from Xiamen University in China as well. And so our target is to address the SDG, the goal 14.1, to prevent and significantly reduce marine pollution of all kinds by 2050, okay? So our aim is to review the situation, identify the pollution hotspot, and also what are the priority pollutants for control, okay? And then we also develop solution together, okay? Today we will have a chance to raise out some idea for different socio-economical status, what that countries can do to reduce pollution. Then we can have a tailor-made solution being recommended to deal with this problem. So the slide is too small, but I can tell you the blue one means we will develop the standard method, then do the sampling campaign together, and then we will do the analysis. The green one means we want the risk assessment. We have to conduct the toxicity test and then find out the effect threshold for each chemicals we target to, and then we work out the risk assessment, identify the risk in different estuaries for global comparison and for management. Then after that, we will have meetings, stakeholders meeting with UNEP and also country representative to try to solve the problem. So here will be our expected outcome. We will form this global network for pollution monitoring, and then we also together develop some new technology for monitoring like passive sampler. We will have this very strong capacity building for any similar types of project. We can work together as a team. We will share all the data, and then for the management, co-design solution, and also we will provide the data to inform decision making. Finally, after the 10 years program, hopefully we can make progress towards the cleaner and safer ocean. Okay, that's our hope. I think this is a really exciting project. Without your contribution, we never achieve this goal. But now we work together, we'll make a step forward. So it's very romantic in a way. So now we are here. We launched the program, sampling program last July, and then hopefully we will finish all the sampling by this summer, and then we will finish the analysis, chemical analysis by the end of this year. Then we will publish the paper together in 2025, hopefully summertime. And now move on to the main body for today's discussion about the next phase of the program. The idea is that through the discussion, some of you will be interested to be the leader, like maybe you're interested to lead heavy metal study. It's okay. Then you find the resources. We will help you through this platform, people sample for you. Then you do the analysis. For example, we want more leader emerging from this room, and then you can take charge of some program. We are not saying all the programs should involve 150 estuaries, too expensive. Maybe you can have a smaller program, say each continent, 5 estuaries, okay? That means only 30 estuaries you can deal with. So we can have different types of projects. Hopefully, through the discussion today, then you indicate your interest, we can have a more in-depth discussion with you, okay? So, for example, here, we suggest possibly we can look at other contaminants of emerging concerns like PFAS, other film retardants, plastic sizes, and also microplastic, pathogens, antibiotic resistant genes, and even look at biodiversity using eDNA. Okay, so we will discuss about this kind of option today. Now, the next phase, since we will target to certain group of pollutants, we need more sophisticated sampling device. For example, Professor Rudolph, who also in this conference, he and I developed this artificial muscles to monitor heavy metals in the past. Now we modify the chelating agent being replaced with a new polymer. The polymer can capture metals and also radionuclides. I will explain to you in a minute. We're going to deploy this around the world with your support. Then we will make hundred thousands of these artificial muscle and send to you. If you're interested, if you have the capability to deploy them into the S3 for 1 month, we need to deploy them for 1 month to accumulate enough of those metals and radionuclide. Then you return them to us, we will do the analysis. Then if you're interested, again, we will send out a questionnaire, you just tell us yes, then we will send to you and start the second phase of project like this. Another possibility is to use this DGT, organic DGT device and other absorbent to look at other target persistent organic pollutants. Then final one is easier one. We will invite you to collect sediment and also oyster. Because oyster is an estuary species. There may be regulation not allow you to send the oyster, but we find a way. You can ask the seafood trading company to send the samples to us for consumption. Easy, that one can be done. So we will find some way that to collect your oyster, either frozen one or live one. Now, this one is example. We've done the work all the way from Japan to Singapore. We collect the oyster to look at pharmaceuticals. Then we find very high level of antihistamine. We just published very recently being selected as fund cover by ESMT. Now, the challenging issue is that we have to do risk assessment. That means we need two kinds of information: the environmental concentration and also the predicted no effect concentration. Means the effect threshold, how much we can discharge without affecting the ecosystem or the marine organism. So without this PNEC, we cannot proceed to risk assessment. What we find so far, we look at just an example in Greater Bay areas in China, southern China. Then we look at the 20 most commonly occur new chemicals pollutant. Then we find 62% of them without PNEC. That means we need to do toxicity tests to find out the PNEC for risk assessment. So in this GM program, we're going to have a lot of unknown, the chemicals without toxicological information. Then I now apply for resources to work on this so that in the downstream risk assessment, we can do a proper job. And now I spend another maybe 5 minutes to talk about this artificial muscle on behalf of Professor Rudolph Wieseko. This is a new development. The old design only for capturing metals, but due to the discharge of radionuclear contaminated wastewater by Japanese government, then we have to increase our ability to monitor the safety of our ocean. So currently, we can use this method filtering with this kind of RAD disk to gather the radionuclide for measurement. However, it has a problem because radionuclide in low concentration in the environment, you need a lot of water sample, at least 500 liters of water sample for filtering. Okay, that is time consuming and also not easy. So then we try to bring about our well-developed artificial muscles to change this wet scene here. Okay, this artificial muscle has an advantage because it can give a time integration of the pollution level highly sensitive to the change of the metal concentration. Sorry. And also very cheap, only $1 each. And also, we don't need to have a invasive procedure to kill animals, okay? So we done the original artificial muscle monitoring in 27 countries up to now. But now we try to modify it to monitor radionuclide as well. So the idea is we change the polymer so that it can capture uranium, cesium, strontium, and other radionuclides. Then initially, we have this idea using zeolite technology as the binding component. Then finally, we developed this technology. It can capture uranium and cesium and also strontium at the end. However, it cannot capture metals. That means we have to use old polymer and new polymer together, two concepts. So at the end, they also try out this technology in different parts in the Western Pacific. Finally, we have to find something new. One bird killed, one stone killed two birds, okay? So we developed this new chelating agent called IAC, okay? We just got the patent done. And then this diagram looks quite complex. This is controlled without solvent, the level of the metals. The first column is cesium, second column uranium, the last one is strontium. So if you see the reduction, that means this chelating agent absorb the radionuclide. Then you can see IAC perform really well, okay, for all three kinds of radionuclide. So it turned out working nicely, okay, compared to other technology. And how about metals? It did really well as well. If you look at this diagram, all metals can be removed by IAC, our technology. So we're going to send you this new one. We will try together. I remember the first time DGT technology came out, they published in Nature. So we will target something high level. We will work together to showcase these really effective tools to monitor radionuclides and heavy metal together. So now go back to this workshop. There are 4 aims in this workshop. First, you will be provided with an update of our progress, okay? We will co-design the next phase of GEM together. Then we will discuss how we can offer practical solutions to solve the pollution issues around the world. Finally, we will try to initiate more collaboration with other parties like potential donors or private company like Elsevier. So if Elsevier can donate us $1 million would be nice. The progress, the one line is like this. After my talk, I will invite Demi from Nigeria, also my PhD student, talk about the desktop study on the pharmaceutical pollution in estuary. And then another student, Lo Chong, will talk about how did we develop the standard methods for sampling. Finally, most excitingly, our project manager, Dr. Tong Chen, will tell you the preliminary result and also other aspects in relation to our progress. So without, maybe I can answer some questions you may have by now.